Hello, my name is Munya Hamadi, and this summer, with the help of Dr. Ren Guerriero and Matthew Perez, I set out to understand the molecular processes of novel sleep-related genes in fruit flies, also known as Drosophila melanogaster. So what are novel sleep-related genes? Well, there are genes that acquire evidence that shows that they may be somehow associated with the physiological and molecular processes of this vigilant state. Some genes that have been found to play a role in sleep include period and clock, both of which are associated with the circadian rhythm of this vigilant state as well as the sleep-wake cycle in general. There are 3 novel sleep-related genes that are of interest for this experiment: the straitjacket, the PTX1, and spinophyllin. These 3 genes can be found in the Drosophila melanogaster, also known as the fruit fly. The main reason that we've decided to use the fruit flies for this experiment is due to the plethora of data behind this model organism in regards to genetic research, and due to the fact that their genes are orthologs to that of mammalian organisms, such as us and house mice. In house mice, they do not acquire the straitjacket gene, but they acquire a gene that is an ortholog to that, also known as CACNA2D3. An ortholog, or an orthologous gene, is one that descends from a common ancestor. Thus, not only are the flies inexpensive to maintain and acquire a very simple genetic structure, they also follow the 6 conditions of sleep. The first one is behavioral quiescence, where it shows inactivity or dormancy. The second is homeostatic regulation, with their ability to make up for sleep that has been lost if they've been sleep deprived. Third is circadian control, where even if put in light conditions or dark conditions, they still follow a cycle for sleep and wake. Fourth is arousal threshold, which is reduced responsiveness to stimuli. Fifth is reversibility, where with a large enough stimuli, they are able to be woken up, unlike in a coma or a vegetative state. And then 6th is posture, where they acquire a distinct posture with their heads down and their wings down. Thus, with all this background information, this summer I went out to see if these novel sleep-related genes were indeed novel sleep-related genes, and if so, what were their molecular processes. I hypothesized that the genes are in fact associated with sleep somehow. When we look at the straitjacket's ortholog in mice, we see that it is a calcium channel protein that interacts with circadian genes that controls the mice's internal clock. Thus, when the gene is disturbed, they acquired a reduction in sleep overall. Preliminary data shows something similar with the spinophyllin gene and the PTX1 gene in the Drosophila melanogaster, but instead of seeing a decrease in sleep, we see an increase in sleep in both sexes, males and females. But in order to begin experimentation, we needed to acquire a large stock of flies. Thus, we made a plethora of food that was molasses mixed with propionic acid. The propionic acid was added to mitigate the risk of contamination from bacteria or fungi, as well as constantly flipping the vials to separate the generations and keep overcrowding to a minimum. The flies were housed in an incubator kept at 21 degrees Celsius and put at 12 hours light and 12 hours dark conditions. To understand the sleeping habits of our flies, we ran 3 trials with the Drosophila activity monitor. If the flies did not cross the beam within 5 minutes, it was dictated as sleep. Each monitor held 32 flies, each within 5 millimeter tubes. The data that we will acquire will be able to be drawn as a line graph that will show the trend of sleep over time. Thus, I have developed a code that takes the raw data from the DAM2 monitors and puts them into the RStudio system, thus outputting the results as a line graph that is easy to read and easy to follow. The pandemic has made it rather difficult for us to pursue with our experimentation due to the constant shipment delays and the back orders. We've only now acquired our CO2 that separates the virgin females from the males that can then be utilized to cross the proper genes that will be silenced or reduced. Nevertheless, we've begun to collect a plethora of data on our parental lines and hope to continue to move forward. With the trials currently being run, with the code functioning, and the plethora of stocks that we have acquired, I would like to thank my coworkers for assisting me with all that we have done so far, the interdisciplinary program of neuroscience at George Mason University, Dr. Ren Guerriero's lab for taking me in to pursue this project, and the Oscar URSP for providing me with the funding and support to do this throughout the summer. Thank you for taking the time to watch my presentation. Hopefully in the next few semesters we will acquire some more concrete data on these novel sleep-related genes as well as their molecular processes in the Drosophila melanogaster. I appreciate your time.